Hello, everybody. My name is Giulia, and I come from the University of Milano Bicocca in Italy. I'm really happy to be here to present my work about association room mining to investigate microbiome patterns, guidelines, and user-friendly tools for real application. So association room mining is a data mining technique born in the 90s for transactional data analysis, so to predict customer behavior and ameliorate selling strategies. The goal of this technique is to find patterns and association between co-occurring elements. And it is a really versatile technique that can analyze very different types of information. And also we have several metrics to explore patterns with specific characteristics. With this work, I would like to merge the two words, so the 16S microbiome data and association room mining to find microbiome patterns in our dataset. So 16S microbiome data are quite a standard in nowadays, and we have a lot of data sets. There is also an increasing use of machine learning procedure and a deep interest in pattern detection, in particular related to environmental variables. And association room mining is a supervised machine learning technique that analyze co-occurring elements, so presence, absence data, and can generate a lot of data, but in particular patterns of any length. So on the contrary, it can produce a lot of data, often also difficult to visualize, and can need computational efforts. At this moment, there is no assessments about ARM and microbiome data, but I would like to introduce this kind of assessment and try to show you how we can image the application of ARM for microbiome data with an implementation of a library, a Python library, to directly use this kind of technique on microbiome data specifically. So this is a little example where we have our presence absence table, and ARM, it can be divided in two principal steps: the patterns calculation, so the calculation of the group of species, in our case, that co-occur based on a threshold. So the support or the frequency is the threshold that algorithm used to calculate all the item sets, all the combination of our item sets with a higher or equal support. So it is the frequency at which we are interested to find some patterns. And then the second phase of the ARM is finding associations, so rules between this kind of patterns. But we can use ARM in several ways. So we can use ARM to find very common patterns, so with a high frequency, but also with a lower frequency, maybe related to environmental variables, thinking to our case studies. So considering the type of item sets, we don't only have a group of items that co-occur, but also other two types that are closed item sets and maximal item sets. An item set is closed if there exists no superset that has the same support count as this original item set. And maximal item set, an item set is maximal frequent, is none of its immediate superset is frequent. So it's like a matryoshka where the maximal characteristics is when patterns are really common in our data set, so really strong associated. And this has really different execution time for the calculation, and also size of dimension, and also the number of item sets that we can generate. Consider these three types of item sets are really different. So considering 1000 matrices with these characteristics, so 100 species and 50 samples, we can generate a lot of item sets, but the maximal are the lowest. And this is also, these characteristics is the same considering the execution time and the dimensions. But of course, using this kind of item sets, we can lose some information about our data set. And for this reason, it can be useful to calculate the item sets globally, and then filter them with some interest measures. In particular, I reported here all confidence and cross-support. For example, cross-support exclude patterns in which high-frequent items co-occur with low ones. So this kind of measure can avoid spurious information, thinking about our microbiomes. If a microbes is really high frequent and another is really low frequent, the association between them, it can be only related to the highest, with the highest frequency. And so this is not informative. So maybe cleaning the information with this measure can be useful for us. Here I reported an example with the ECAM dataset that is published in 2016, where we have 2 years monitoring of microbiome babies, collecting information about diet type, antibiotics administration, type of delivery, and also sex. And what I would like to highlight is that if we use different input tables, so considering different taxonomic levels and considering different support threshold, we can have very different amount of information. Also with numbers that are reliable for our type of analysis. So thousands, less than 10,000 is quite similar at the ESB that we can calculate during a microbiome analysis. But on the other hand, we can calculate item sets also considering different type of inputs based on the metadata. So we can filter the data sets based on the metadata and then calculate our patterns, and we can have very different results, and we can reach patterns with a lower frequency in this way. And also we can include the metadata into our analysis with the option yes or no into our patterns, and this can be really useful. And we can use also metrics to remove spurious information. As I said before, this is the analysis considering the cutting of spurious patterns with the all confidence metrics. So for this reason, I decided to implement a Python library that use the common microbiome inputs and then calculate the item sets and perform some similar microbiome analysis with the patterns. So the taxa table, so where we have abundances for each sample, are converted into list of transactions. And then we have the calculation of frequent item sets using the parameters, and we have template files to simplify this procedure. Then we have the additional metrics, and then we have also the inclusion of metadata with the conversion to a pattern table. So we don't have a not view table, but a pattern table with the presence absence of each pattern in our samples. And then we can reach a biodiversity analysis and a visualization with an interactive dashboard. So this is the nucleus of the tool, the library with the Python modules to importing data, managing and filter files, calculate item sets, include additional metrics, perform microbiome analysis with patterns and plots. We can use the library using the function if we code, but also we can avoid the command line interface using the interactive dashboard that are implemented into the tool, and also the guided scripts if we want to use the scripts that are pre-settled for an easy use. So there are different ways to use them. This is an example of the dashboard. It's a copy and paste because the space is this, but we have the filters. So we can filter our pattern table with the metrics of the analysis, but also with the metadata of our samples. And then we can visualize interactively with the common plots of biodiversity or microbiome analysis, like heat maps, for example. This is the pattern table, so a mixing of standard representation of OTU table with patterns. So we have patterns with presence and abscess for each samples, and then columns with our metrics to filter the patterns. So concluding, the library, it's a library implemented in Python that try to meet the needs of microbiome for the application of ARM. And it can generate interactive dashboard to explore dynamically microbiome data and the inclusion of the metadata into the analysis. And it can provide some guidelines to set up the ARM analysis, and also it can introduce the pattern table. My future work, of course, are carrying out the assessment of 16S and ARM, but also supporting high-dimensional data and a step-by-step library implementation considering microbiome characteristics. And of course, I hope in a new application of machine learning approach to microbiome data. So I would like to thank you, of course, but also to thank the conference organizers that really helped me in these days, and the International Society for Computational Biology. And I would like to thank my university and the Zooplant Lab group, and also my colleagues of the TechSpeed PhD program. Thank you so much.